Hello, and welcome to the Cancer Research Institute Cancer Immunotherapy in You patient webinar series. Today is Wednesday, August 15th, 2018, and the title of today's webinar is Biomarkers and Cancer Immunotherapy, What Patients Need to Know. Before we begin, I'd like to thank our generous sponsors who have made this webinar series possible. Bristol Myers Squibb, with additional support from Regeneron, Sanofi Genzyme, and Adaptimmune. My name is Arthur Brodsky, and I'm the science writer at the Cancer Research Institute, a nonprofit organization established 65 years ago with a mission to save more lives by funding research that aims to harness the immune system's power to conquer all cancers. This work has contributed to the development of life-saving immunotherapies for a variety of cancer types. We present this webinar series to patients and caregivers to help them understand what immunotherapy is and how it differs from other treatments, to provide information on the latest developments in research and treatment, and to connect patients to immunotherapy clinical trials. Now, it is my pleasure to introduce today's expert speaker. Dr. Kunle Odunsi is currently the Cancer Center Deputy Director, the Chair of the Department of Gynecologic Oncology, the M. Stephen Piver Professor of Gynecologic Oncology, and the Executive Director of the Center for Immunotherapy at the Roswell Park Comprehensive Cancer Center. Additionally, he is a professor of gynecology and obstetrics at the Jacobs School of Medicine and Biomedical Sciences at the University at Buffalo. As a recognized leader in the field of ovarian cancer immunotherapy, Dr. Odunsi has led several clinical trials involving novel immune-based approaches such as vaccines, while his lab focuses on the molecular characterization of tumor antigens in ovarian cancer and refining strategies to target them more effectively. Dr. Odunsi, it's an honor to have you with us here today.
Well, thank you, Arthur, for that introduction. And I'm going to be focusing on biomarkers. How can we use biomarkers in cancer immunotherapy? As we know, immunotherapy is currently it's really the wave of the present, and it's going to be the wave of the future in the way we treat cancer patients. It's making a tremendous impact on outcome of cancer patients, and cancers that we previously used to treat with different kinds of chemotherapies. Many of them are really sensitive to immunotherapies. And the question is, how can we even improve on our current results? Because clearly there are patients who respond very well, and there are patients who do not do as well. So biomarkers is one potential answer to how we can improve on the results. So what are biomarkers? These are factors that you can measure that can help you to, that can help us to shed light on our health. So, for example, it can help to improve how care is administered, how patients are selected for different kinds of immunotherapies. So, in cancer immunotherapies, biomarkers can actually reveal information about the tumor itself, as well as how the immune system is interacting with the tumor, as well as other important factors. The more we understand the nature of the immune system in relation to tumor, the more we're able to make informed decisions about the best kind of immunotherapy for a particular patient. Because there are different types of immunotherapies, not all of them work equally. They work via several mechanisms. And some immunotherapy may be good for one patient and not the other patient. So biomarkers are very important. So when can you use biomarkers? So if we have good biomarkers, obviously we can use it before treatment so that we can select which patients are likely to benefit from from a particular therapy. So the biomarker can be used to better get information about the type and stage of a cancer. We can look at immune profile within the cancer itself and immune profile in the blood of a patient. We can also answer the question, what is the potential expected outlook for a particular patient based on the biomarker? And as I said earlier on, what is the best treatment option for this particular patient? Moreover, while a patient is on treatment and even after treatment, one can look at additional biomarkers. Some of them, many of us are familiar with. These are things like even CAT scan, a simple CAT scan, or what are called imaging studies, or MRI, or PET scan. Those actually can tell you whether or not a tumor is shrinking and whether or not there's a need to change treatment. In addition, some biomarkers can help us understand the potential for side effects, which patients are more likely to suffer side effects, and in which patients are the side effects likely to persist, which patients are likely to be in sustained remission compared with patients where there's a high probability that the cancer might come back. So biomarkers can be used in so many different kinds of ways. But the question is, how do you measure these biomarkers? How can we identify biomarkers? This is a complex question because it requires very detailed, meticulous studies that usually typically involve a fairly large number of patients. Because you want to make sure you come up with an ideal biomarker that is sensitive. In other words, when it says it's positive, it is truly positive. And when it says, the result is negative, it is truly negative. We don't want to cause any false alarms, and we don't want to cause false reassurance when in fact there's a problem. So there are different ways of trying to measure these biomarkers. One way is to measure directly in the tumor tissue samples, because that is where the battle is taking place between the immune system on the one hand and the cancer cells on the other hand. So it remains the gold standard to look at the tumor tissue samples in order to have a comprehensive view of what is going on in the interaction between the immune system, a patient's immune system, as well as and the tumor. The challenge, however, is that this sometimes require multiple biopsies, which obviously create a source of discomfort for patients. So gaining access to tumor tissues is actually a scarce resource that needs to be considered very carefully within the context of clinical trials. We've talked a little bit about imaging studies such as CAT scans and PET scans. This can help us analyze how the tumor is responding to treatment. In fact, there are some forms of CAT scans that can also measure the metabolism within the tumor. In other words, how is the tumor utilizing, let's say, glucose or amino acids or fat? Because again, tumor cells are like living cells that need nutrition for their survival. And so the immune cells that are within the tumor, they also need the same kinds of nutrition. And sometimes there's a competition between the cancer cells and the immune cells. And the ability to measure this may also give some indication about how a patient is likely to respond. The other type of biomarker that one can visualize in some of the imaging studies is really the degree of immune cell infiltration. But the easiest biomarkers that does not involve tumor biopsies are blood-based biomarkers. Unfortunately, while these are ideal, as I indicated earlier on, it's really at the tumor tissue that a lot of activity is going on, and there are ongoing studies for us to be able to identify blood-based biomarkers that will tell us what is going on within the tumor tissue. Examples include trying to identify circulating tumor cells in the blood, or some of the tumor DNA in the blood, and even the immune cells in the blood, because after all, there are large numbers of immune cells in the blood that we can also analyze. There are some alternative sources of biomarkers, and some of them are beginning to be deployed in the clinic. Many of them are still investigational. So for example, the measure of our gut microbiome from the stool. Microbiome simply means the bacteria that are resident within our gastrointestinal system. Interestingly, this contributes to how a patient responds or not respond to some forms of immunotherapy. So let me begin to talk about some specific biomarkers. I think we already mentioned some of them, but the ones that many of us are probably familiar with are listed on this slide. So there is a molecule called PD-L1 that is expressed on tumor cells. So this molecule is actually a molecule that we block when we use some of the immunotherapy agents, including they're called PD-1 inhibitors. Examples include nivolumab, Keytruda, avelumab. So there are several examples of these molecules that block, several examples of drugs, immunotherapies, that block this PD-L1 expression on tumor cells. And I think in the next slide, I'm going to talk about how this pathway works. Why is it important to block PD-L1 expression? So you can imagine if you're going to block PD-L1, you need to understand whether PD-L1 is expressed in the tumor. So PD-L1 expression could be an important biomarker, and I will speak to that a little bit later. Then the next one that is very obvious is, are there immune cells within the tumor? So you can simply take a tumor biopsy or take a piece of the tumor and ask the question whether or not there is pre-existing immunity as evidenced by presence of immune cells within the tumor. And there are several studies, including studies in ovarian cancer, that have shown that patients whose tumors have high numbers, high frequencies of these immune cells tend to do better compared with patients that have low numbers of immune cells. Another form of biomarker that is gaining increasing attention is the identity of the tumor itself. What is the tumor DNA? What does it look like? It turns out that tumors that have higher mutations in the DNA tend to respond better to immunotherapy compared with tumors that have lower numbers of mutations within their DNA. And then the microbiome is what I mentioned earlier on. This is really the bacterial composition of the gut or of the mouth or other cavities in the body. It turns out that the composition of bacteria within the gut especially affects how we respond to the immune system. And the explanation for this is a little bit complex, but the bacteria appear to send signals. The bacteria within our gut appears to send signals to the immune system about how well they should respond to certain forms of immunotherapy. So let me drill down a little bit more on this PD-1, PD-L1, because there are so many types of immunotherapies that are currently being used in patients that focus on blocking this PD-L1, PD-1 pathway. As you can see from this slide, PD-1 is a molecule that is expressed on the immune cell. This is the killer T cell. PD-L1 is the molecule that is expressed on the cancer cell. When you have engagement of PD-1 with PD-L1, as shown here, what happens is this sends a signal. It puts a brake on the immune cell, puts a brake on the T cell so that the T cell stops working. So basically, this is what happens. So you have the cancer cell sending out this signal of PD-L1 to PD-1, and this essentially abrogates the ability of the T cells to do their job. Consequently, if you block this pathway, if you block this PD-1, PD-L1 interaction, you can rescue these immune cells and make them to become functional. So there has been several FDA approvals of this approach, whereby a lot of immunotherapy agents were approved in the last few years that is focused on this pathway. These include immunotherapy for metastatic melanoma, non-small cell lung cancer, liver cancer, bladder cancer, kidney cancer. I mean, so many types of cancers that it is amazing how a lot of patients that previously had no hope are now able to respond to this immunotherapy, this PD-1, PD-L1 blocking therapy that rescues immune cells and allow them to function and better able to attack the cancer cells. But as I said, not all patients respond very well. So one potential biomarker is to ask the question, is PD-L1 expressed in the cancer? Are those patients likely to respond better? And the answer to that is yes and no, as I will talk about later on. Then if you look at patients with pre-existing immunity, whereby you simply look under the microscope and ask the question, are there immune cells that are already infiltrating the cancer? Are they present in large numbers? And again, the answer is when they are present in large numbers, those patients tend to do better, number one. And number two, those patients are more likely to respond to the PD-1, PD-L1 checkpoint immunotherapy. Because remember, the goal of this PD-1, PD-L1 checkpoint immunotherapy is to block that interaction and rescue already existing cells, already existing immune cells within the tumor compartment. And the next one is the tumor mutation burden. Okay, the tumor DNA plays a role in determining whether or not a patient is going to respond, because what happens is when you have large mutations and a high frequency of mutations in the tumor DNA, this makes the immune system to see the tumor as more different from self. And consequently, it sees the immune system, it sees the cancer almost as a foreign pathogen, almost like a bacteria or virus that is different from other normal cells of the body. So patients who have highly mutated tumors tend to have more of those immune cells, the T cells, and therefore they are more likely to respond to PD-1, PD-L1 checkpoint immunotherapy. And very important to mention here is the historic immunotherapy approval by the FDA for patients who have a certain type of mutations in their tumors. These are called MSH high tumors. Let me just, so MSH stands for microsatellite high tumor, whereby there's microsatellite instability. These type of solid tumors can affect actually almost any type of cancer, but they are typically found with some colorectal cancers, uterine cancers, and some other types of cancers. So these tumors have high mutation burden, and therefore they are likely to have high infiltration by immune cells. Therefore, they are likely to respond better to immune checkpoint therapy blocking PD-1, PD-L1, because now what you only need to do is to rescue those immune cells so that they can function and do their job. So moving on to the next slide. So really the goal is to make sure we come up with targeted approaches so that each tumor, because again, as you can tell from everything we've said so far, each tumor can be different. One patient may respond very well and the other may not respond as well. And these are some of the reasons. It depends on what we are targeting. So a patient that has highly mutated tumor where the DNA has high numbers of mutations, therefore that kind of patient will be able to generate patient-specific T cells, immune cells that are targeted at the mutations in that particular tumor. Some cancers are due to viruses, Two examples are human papilloma virus that is associated with cancers of the cervix, cancers of the head and neck, and some cancers of the oropharynx. So you can imagine we might be able to target these cancers by targeting aspects of the papilloma virus. Another example is the Epstein-Barr virus that is associated with some forms of lymphoma. Then a cancer may also express some additional targets, some proteins that are specific and unique to the cancers, and some of them are listed here and include HER2/neu, which is very familiar to, which is highly expressed in some types of breast cancers. And I want to point out the MAGE family and NY-ESO1. These belong to the family of what we call cancer testis antigens. These proteins are not expressed by normal tissues, but they become expressed. They're expressed in cancers, and the only normal tissue that tends to express these proteins is the adult male testes. So you can imagine that they are very specific because they're expressed only in cancer, but not by other normal tissues. So one can think about targeting tumors specifically based on these different biomarkers. In fact, we can call them biomarkers. When you have expression of NY-ESO1, for example, by a tumor, then we can focus the immune reaction against NY-ESO1 for that particular patient. If there is if viruses are involved, we can focus on the viruses. And if the tumor has a lot of mutated antigens, we can focus on those as well. So I think I already talked about NY-ESO1. It's expressed by different types of cancers, multiple cancer types. It's not expressed in normal tissues except for the testes. For ovarian cancer, tumors that express NY-ESO1 appear to be a little bit more aggressive. And we and others have shown that if we vaccinate these patients, when we focus the immune response against NY-ESO1, we have preliminary evidence suggesting that the patients who have immune responses against NY-ESO1 tend to do better in terms of their clinical outcome. So with regards to the microbiome, this is also another marker whereby, again, depending on the composition of the bacteria in the gut or other body cavities such as the mouth, this can affect how well we respond to immunotherapy. So some types of species of bacteria have been, and bacterial signature have been found to be associated with response to treatment and even the development of cancers of the colon and rectum. When you think about diversity of bacteria in the gut and you modulate the composition of bacteria, especially in some of the experiments in the lab, there's evidence that this can affect response to checkpoint inhibitors. And there are several studies now showing that, the use of antibiotics or the type of diet can affect the composition of the gut bacteria, and this in turn can affect response to checkpoint inhibitors. So going forward, the question is, what are some of the opportunities and what are some of the challenges? A biomarker has to be specific and has to be sensitive. In other words, reduce the possibility of false alarm and reduce the possibility of falsely reassuring a patient about the value of the biomarker. So going forward, we need to continue to work on discovering and validating new robust biomarkers, look for alternative sources of tissue for analysis. I've talked about some of the challenges of gaining access to the tumor specimen itself, because oftentimes that requires undergoing biopsies. We clearly need to develop biomarker panels that will improve the predictive power of immunotherapy. It is entirely possible that not one biomarker is going to do it. Maybe we need a panel of biomarkers and put them together before using them for their predictive ability. We also need to design clinical trials more efficiently and rationally by really being smart about how we design the clinical trials and putting patients on clinical trials potentially based on these biomarkers. So if a patient has let's say PD-L1 expression, for example, if the tumor is full of PD-L1, it makes sense to triage that patient to a clinical trial that has a PD-L1, PD-1 immune checkpoint inhibitor therapy. And if the tumor has some other type of biomarker, to triage the patient to that appropriate clinical trial. And with that, I think I will stop here and hand back over to Arthur.
There we go. Thank you so much for your very interesting overview of biomarkers, Dr. Odunsi. I learned a good bit, and I hope that everyone else that's watching does as well. So now we have a little some time for some questions where hopefully we can delve into some more of the complexities that you alluded to earlier. So first, you mentioned the historic immunotherapy approval involving the MSI-high biomarker. And I thought that was pretty cool because it shows how a single biomarker can be useful for patients with a wide range of tumor types. Are there likely to be more biomarkers like this, or do you think that most will only be relevant in specific cancers?
The short answer is, I think the short answer is going to be yes. There are a lot of ongoing studies looking at biomarkers that will cut across multiple cancers. Every type of cancer is different. For example, a cancer of the ovary is different from cancer of the uterus, and they have some unique properties that are distinct for the particular cancer type. Nevertheless, there are some general properties across cancer types that one can utilize in order to develop biomarkers that can be applicable to more than one type of cancer. So in my view, I think we're likely to see more of that type of example, such as the microsatellite high tumors that cuts across multiple cancer types, and with the understanding that those patients tend to respond better to checkpoint immunotherapy.
That's good to know. It offers hope that we don't necessarily need to treat cancer in that one-size-fits-all, or rather this could help improve that one-size-fits-all manner and hopefully enable better strategies across cancer types like you mentioned. So next question, you highlighted the PD-L1 biomarker's value, especially when it comes to immunotherapies that target the PD-1, PD-L1 pathway. But you also alluded to the fact that it's not a perfect predictor of which patients might benefit. Can you talk a little bit more about some of the limitations of biomarkers such as PD-L1 and how they're being addressed in the clinic?
Yeah. So the PD-L1 biomarker is a perfect example of where you need to have biomarkers that are robust, sensitive, specific, and gives you good predictive value. And the reason I say that is that clearly patients with PD-L1 expressing tumors, a lot of them, a significant proportion, will respond to immune checkpoint immunotherapy when you are blocking PD-L1. However, there are patients whose tumors do not express PD-L1 who respond just as well. That's number one. Number two, the expression of PD-L1 appears to be dynamic, whereby it can change from time to time based on some of the other factors that are going on within the tumor microenvironment. So if you measure it today, it can be negative. Maybe it's positive tomorrow based on some of the other things going on within the tumor. Number three, PD-L1 expression, generally, it's not universal within the tumor. So if I look at the tumor under the microscope and I stain for PD-L1, not every cancer cell within that tumor is going to be PD-L1 positive. So there will be cells that are negative and there are those that are positive. So how do we interpret that? Is that patient going to respond or not? So what is the appropriate cutoff of positivity that allows, that tells us that this patient is likely to respond or not? So if you look at the literature, there has been several thresholds of positivity that has been used. And so it is not entirely clear what is the appropriate threshold. This is an area of ongoing research, but I think the PD-L1 expression as a biomarker is an interesting example of how much progress we've made, how much we know, and how much we still don't know, because it is not a perfect biomarker. I have to say that PD-L1 expression as a biomarker has been approved by the FDA to accompany some of the immunotherapies. So it is useful, but I think we need to do more work to find more reliable biomarkers.
Makes sense. So you kind of touched on portions of the answer to the next, or presumable answer to the next question I'm about to ask. But in general, what makes something a good biomarker?
So there are a number of parameters for a good biomarker, and I think I alluded to some of the parameters during the presentation, and they are really as follows. Number one, what is the if the question is this biomarker predicting patients who are likely to respond, I will ask question about the true positive rates. When you say it's positive, when you say PD-L1 expression, for example, correlates with patients who respond, what is the true positive? Then number two is, what is the true negative? In other words, when it is negative, how well does it predict that a patient is not going to respond? Then I ask the question about what is the false positive rate? Okay, when I say PD-L1 is positive, therefore I expect the patient to respond, and the patient doesn't respond. That is a false positive result. And then finally, what is the false negative result? So those are all components that we put together in order to arrive at the predictive value, reliability, and accuracy of a biomarker test. And those are very important considerations before we begin to, we really need to understand the reliability of a test. When I sit in front of a patient to talk about the use of immunotherapy, I want to have as much confidence as possible in the test, because it affects decision making about what kind of immunotherapy, what is the expectation, because the last thing we want to do is to give false hope. We want to be able to manage expectations appropriately.
Mm-hmm. So one part of your talk that I found really interesting was your discussion of the bacteria within us and how their effects on the immune system and on cancer, and especially the part about how patients that have the more diverse bacteria are more likely to benefit from immunotherapy. So first, how could these bacteria-related considerations impact treatment decisions for doctors and patients? And then second, how soon do you think we'll start to see some of these bacteria-related biomarkers incorporated into common clinical practice, if indeed they do turn out to be good biomarkers?
Yeah, so that is actually one of a very remarkable finding over the last few years by several groups around the world. The role of, who could have thought that bacteria residing in the gut will have an impact on how you respond to immunotherapy? We don't fully understand the explanation yet, but part of the explanation, at least, is that these bacteria shape the repertoire of immune cells that we have in our body. So depending on the kind of bacteria, it plays a role in how we then respond to other things, including how we respond to cancer. Moreover, some of the bacterial cell wall properties, some of the properties of bacterial cell wall, when they become released into the blood have some effect in modulating immune reaction. So the complete explanation is not clear. There are several lines of thought about how it works, but the point is the bacterial composition affects our ability to mount an immune attack against cancer. So the question is, how do we now use the information? There are several ongoing studies, especially in the context of clinical trials, where we are carefully monitoring and measuring and analyzing the bacterial composition of the gut. In our case, for example, at Roswell Park, we are analyzing the microbiome, not only from the gut, but also from the skin, from the vagina, from the oral cavity, trying to understand the impact of bacteria from all of these areas of the body's anatomy. How do they shape immune response to cancer? I think we still have a long way to go in trying to figure out what is so one question is, what is the best composition of bacteria to have? Okay, what are good bacteria? What are bad bacteria? Does it mean if there's a bad bacteria that's likely to negatively affect the ability to respond to immunotherapy? Does it mean we try and eliminate those bad bacteria? Are there strategies to alter the composition of the bacteria, the gut bacteria? So there are all kinds of questions, and many of these are being explored, both at the preclinical in the laboratory, as well as part of some ongoing clinical trials. So I'm hoping within the next year or two, we will have more information about how to use that information and use it actually, maybe even to develop a biomarker as part of a biomarker panel in figuring out which patient is likely to respond based on the bacterial composition. And if it turns out that you are not likely to respond, how can we manipulate the bacterial composition to enhance the chances of success with immunotherapy?
Great. I want to follow up on one thing you mentioned there. You mentioned how a lot of these questions about biomarkers with respect to the microbiome, but also all biomarkers, that clinical trials are very important for answering those questions. Could you just talk a little bit about the role of clinical trials in helping to advance the science of immunotherapy?
Yeah. So clinical trials are very critical. In fact, we will not be where we are today without clinical trials. The success that we talk about with immunotherapy is based on clinical trials conducted over the years by several investigators around the world. And I think we owe a debt of gratitude to not just to the investigators that have persevered, but also to patients that participate in these clinical trials. One important point to make at this juncture is that the way we design clinical trials nowadays are different from 20, 30 years ago. Clinical trials are not designed just as a last option. They are designed, in fact, some of them, even though some of them are called phase 1 trials, these trials are designed many times, not just because you're looking at safety in patients who are endpoint. In fact, nowadays, phase 1 trials are designed looking for signals of efficacy, signals of potential for cure. So if you are listening to this, I'm encouraging participation in clinical trials, because they're designed with intent to provide benefit. I think I can say most clinical trials are designed with the intent to provide, actually I'm focused on phase 1, early clinical trials. They are designed with the intent to provide benefit, not just to look for safety and toxicity, which is the traditional paradigm 20, 30 years ago. But importantly, when participating in the clinical trial, the ability to obtain tumor biopsies to really understand what is going on in the battlefield. How are the immune cells interacting with the cancer cells? How are they changing in response to therapy? I think it's very critical to understand this because, again, that is how we got to where we are. By studying this in detail, understanding, why patients who are responding, and understanding why those who are not responding are not, so that, we can come up with other strategies.
I completely agree. I think I myself was even a bit surprised once. There's a lot of myths out there, I think, about clinical trials, and it's reassuring to know that they are much more helpful, I think, than a lot of people realize. So we're almost out of time, but I've got one last question. So even with all the remarkable progress that's been made with respect to biomarkers recently, as you touched on, it still seems like there are many more advances ahead that will help doctors better understand the complex relationship between cancer and the immune system, and in turn that would enable them to provide their patients with even better care. So before we wrap things up, I was hoping you could provide your vision for the future of biomarkers and how doctors are really working to take advantage of their full potential to improve patient care.
Yeah, I think I have to say that the future of biomarkers, the future is very bright. I think in a few years from now, we will have a lot of answers to the remaining open questions. And the reason I'm very optimistic is because of advances in technologies. So, for example, right now, it's possible for us to rapidly take a tumor specimen and sequence all of the 30,000 genes in the human genome. So you can understand exactly what is the tumor DNA doing. So technologies and advances in ability to really understand what the tumor is doing, what the immune cells are doing. There are technologies to analyze the immune cells at a depth that is totally unimaginable even only about 5 years ago. So with all of those tools in hand, there are already a lot of studies, especially studies associated with clinical trials, that are going to lead to discovery of either new biomarkers or panel of biomarkers that we can use for our patients. The other important technical advance is our ability to put big data together. So there is actually like what is called data science, where we can put multiple types of big data, because as you can imagine, you do these studies where you're looking at the tumor genome, the immune system genome, you're looking at the microbiome, billions of bacteria in the gut, you're looking at the immune cell populations. How do you put all of this data together in order to come up with a biomarker panel? So the field of data science is advancing also very rapidly. So collectively, if you put all of these developments together, I have to say that the future is bright, that it's only a matter of probably a few years. I don't want to, I don't want to put a number, but I think we're accelerating at a very rapid pace towards identifying predictive biomarkers that would tell us exactly what kind of immunotherapy each patient needs, and really be able to personalize immunotherapy the way it should be.
Thank you, Dr. Odunsi. It's great to hear how promising these biomarkers are starting to be for patients and their potential for how much better they can even become. Well, that is all the time that we have for today. Thanks again for your extremely informative webinar. I hope that everyone out there learned a lot and that this information will prove helpful.
Thank you. My pleasure.
For more of our webinars and the additional resources that the Cancer Research Institute has for patients and caregivers, you should check out our Answer to Cancer educational programs. And we encourage you also to check out our website at www.cancerresearch.org. Here, you can read and watch stories shared by others who have received immunotherapy treatment across a wide variety of cancer types. You can register for one of our immunotherapy patient summits, browse our entire library of past webinars featuring the world's leading immunotherapy experts, such as Dr. Odunsi. You can access information on other resources, including treatment, emotional support, and financial assistance, and find help locating an immunotherapy clinical trial. On June 30th, we kicked off our Immunotherapy Patient Summit series this year in San Francisco. At these free half-day events, we connect patients and caregivers with immunotherapy experts to discuss the latest advances in research and treatment. Other summits are scheduled for New York City, San Diego, and Houston. Please go to cancerresearch.org/summit to learn more and register to attend. Finally, I'd like to thank our sponsors one last time for making this webinar series possible: Bristol Myers Squibb, Regeneron, Sanofi Genzyme, and Adaptimmune. I'd also like to thank and recognize BioRender, whose platform was used to create many of the graphics that you saw today. And again, you can watch this and all of our other webinars on our website at cancerresearch.org/webinars to learn more about the immunotherapy advances in a number of cancer types. Dr. Odunsi, I just want to thank you so much again for taking the time with us today and for the amazing work that you are doing to help patients. We wish you the best of luck.
Thank you.
Thank you.